The urea cycle is how the body gets rid of excess ammonia when amino acids are broken down. Before we enter the cycle, let's explore how ammonia is first moved from other amino acids to make glutamate in a process called transamination. So we start with an amino acid that combines with alpha-ketoglutarate, and that leads to some kind of alpha-keto acid, which has this structure, and glutamate. The glutamate goes on to be deaminated in the urea cycle, and the alpha-keto acids are used in a lot of other cell processes. Okay, so now that we are going to start with the process of deamination, let's set the stage. Most of this occurs in the liver, and liver cells are known as hepatocytes. So this is occurring in a hepatocyte. Inside of the hepatocyte, there's also a mitochondria, and some of this happens in the cytosol, some happens in the mitochondria. We enter the mitochondria as glutamate, and that glutamate gets deaminated, or the amino group is removed from the glutamate, making alpha-ketoglutarate and ammonia. Now that we've got our ammonia, the urea cycle can begin. We start with 2 ATP and bicarbonate and ammonia, which is going to combine together to create something called carbamoyl phosphate. This happens in a series of steps that require the use of ATP, and one phosphate group gets attached to the carbamoyl phosphate, but one gets removed as just an inorganic phosphate, and 2 ADP are also produced. Meanwhile, in the cytosol, you've got a molecule called ornithine that was created in the last urea cycle that has to enter the mitochondria to combine with the carbamoyl phosphate and keep it cycling again. The structure of ornithine contains an alpha carbon, and attached to that alpha carbon are an amino group as well as a carboxylic acid. So that makes this an amino acid, although ornithine is not one of the amino acids that's used to make a protein in the human body. Before the ornithine can combine with carbamoyl phosphate, it has to first enter the mitochondria via some kind of transporter. Once it's in the mitochondria, the reaction can occur and citrulline is made, removing that phosphate group in the process. Back in the cytosol, we've got a molecule of aspartate that we're going to have to combine with the citrulline, which is going to take a whole lot of energy. So first up, the citrulline has to leave the mitochondria via another transporter, and it reacts with the aspartate using the energy that comes from both of the phosphate groups from that ATP molecule. This reaction creates a molecule called argininosuccinate, which is just a combination of arginine and succinate. In the next reaction, that succinate is removed in the form of fumarate. Basically, 2 of the hydrogens are removed and added to that nitrogen, and the carbon becomes a double bond. And now we're going to be left with just arginine. Yes, just like the amino acid, arginine. And now we have hit the final step. Water comes in, and urea comes out, completing the cycle.